So, this gives you an example of a polygenic trait. So, the nature of polygenic trait is they are not binary, it is not like tall and short or terminal flower or axial flower, purple or white. So, they have variation, okay, there are shades, like if you take for example the height of human beings, okay, it is not like there are set of people who are dwarf and there are set of people who are tall. So, you have an entire range. If you measure the height of a certain number of individuals, like for example this class is little bit small but still good enough. If you measure the height of all of us and plot it, the graph will look like the one seen in this slide. Okay. So, extremely short or extremely tall individuals will be fewer in the population and majority will be somewhere in between the two. So, there will be an average where the number will be maximum. So, it gives you a bell shaped curve, and that is because of multiple genes each having different alleles, and what combination come together could determine the outcome, phenotypic outcome. So, here we are talking about skin color in some organism, I forgot which one this is. The skin color here is determined by 3 different genes. Okay. So, let us say you have a parent with which is totally heterozygous for all the genes, and then you cross with another one like pretty much selfing, then you will get these many combinations, genotypes, 164th will be completely recessive and so on. And the other extreme again is 164 completely dominant, and then in between these many combinations. So, what is the contribution of a given allele of a gene in combination with another allele of another gene and so on? So, you get variations in the color. So, and that kind of tells variation in the phenotype is usually a good indicator of polygenic inheritance. A human height is a good example, and in this particular organism the color shade is a good example. Is this clear? Polygenic inheritance. Okay. So, now this seal can quickly breeze through. So, environments do influence the the gene, may be a single gene produces a certain pigment, and the pigment color could be sensitive to the pH. So, the plant grown in alkaline soil versus acidic soil can have different colors. This does not mean in this plant flower color inheritance failed to follow Mendelian inheritance. And there are opposite situations. You will get multiple phenotypes due to one single gene mutation. It depends on which gene is affected. A good example is sickle cell anemia. Here you have one amino acid mutation, a glutamic acid to valine mutation due to one base change, but then that affects the 3-dimensional structure of the globin polypeptide such that the hemoglobin molecules can aggregate and form long rod like shapes, aggregations, and that affects the shape of the RBC, and the RBC becomes sickle shaped, and that is why this is called a sickle cell anemia. Okay. So, the genotype is clearly one single variation. So, 2 different alleles, one has glutamic acid, another has valine, 2 different alleles, one single gene only, but the biochemical nature of that protein causes multiple phenotypes due to that RBC being like that, and those RBC are going to be broken down, and because they are not available and oxygen is not transported, then you have problems like physical weakness, anemia, and since they clog the artery, then you have problem on heart, then blood vessels get blocked, and due to that you have problem in brains, and these cells accumulate in organs where these are degraded, and due to that damage to other organs, and as a result the final outcome here, just at least 5 of them are listed. These are 5 different phenotypes coming from single mutation. So, here again you have Mendelian inheritance being followed at the genotype level, but not at the phenotype level, and you understand the phenotype and you understand the biochemistry and physiology of how that particular gene product functions. Okay, so all of this, to learn all of this and to learn lot more, which we will learn in this class and in next few classes, you do not need to know the molecules. Okay, you do not need to know chromosome or DNA sequence. So you can simply generate mutations and set up genetic crosses, follow the inheritance pattern, faithfully adhering to Mendel's laws, then you will learn lot about what those genes do, okay, without knowing even the protein sequence. Like we found a gene involved in garden peak color, right, but we did not find out what protein is, but we clearly know there is a gene that is determining the flower color. So in that same way you can do complex genetic experiments and learn a lot about gene function without really knowing protein product. So then we are kind of going back to history and trying to recollect the historical sequence of events very quickly. So people wondered, fine, based on the phenotype we understand what Mendel is talking about, this gene etc., but what is the physical basis of it, what is the nature of gene, where are they present, you know. So then people who were observing cells using microscope, called cytologists, chromosomes segregate very much like the way Mendel's genes. Okay. So for example, chromosomes and genes both are present in pairs. You have, like for example in our cells you have 23 pairs, meaning there are chromosomes, like for example if you take chromosome 1, there is one more chromosome among the 46 that look like chromosome 1. Like that you can actually sort the 46 into 23 pairs. Okay. So therefore they exist in pairs like Mendel's capital P or small p. And homologous chromosomes separate and alleles segregate during meiosis. Okay, so you get only one of the two chromosomes. I am going to say allelic chromosomes, only one of the allelic chromosomes comes to the gamete, and Mendel's assumption also one of the two capital P's only will be there in a gamete. And fertilization restores the two capital P's together or a capital P with a small p, and similarly the homologous chromosomes come together again. So all of this told that probably genes are on chromosomes. Okay. So you celebrate the solving of double helix, right, but these were the bedrocks to that discovery eventually. So this illustrates in a cartoon fashion what I just told you. So you assume for the flower color it is on one chromosome and shape on the other chromosome, and then you find only one each in the gamete, and we are assuming here, okay, they are in two different chromosomes, and therefore we are only taking two chromosomes here, and fertilization they come together, and then you have different combinations. So this we will see in some detail in the next slide. And then you have this homologous chromosome separation, and then the sister chromatids separate and then they come together to make the next generation. So if you assumed color is on one chromosome and shape is in the another chromosome and follow through this meiotic process, and then you get this gametes, and then if you bring it to the F2, then you will find they follow the same ratio as Mendel's experiments. Okay. And so there is another evidence that conclusively proved that genes are on chromosomes, that came from Morgan's work. So I think I mentioned this earlier, right, while talking about preformation and epigenesis, right. So the Morgan identified, unlike the pea plants where there were lot of characteristics or phenotypes in which variations were naturally available, in Morgan's experimental model Drosophila there weren't variations, so he didn't find any, and after a lot of search he ended up finding just one male that had white eye. Okay. and successfully setting up a mating with that one male, okay, he discovered eventually that genes are on chromosomes. Okay, we will see that just for the fun of it, okay, even if you may already be familiar with this. So, now let us assume, okay, the gene for eye color is on the X chromosome. Now, we will call this W plus, okay, that is how the Drosophila geneticists do. You know, they put a plus for the wild type or the one where the function is like dominant allele, and minus for the one that is mutant or where the gene function is not there. In this particular example, the plus is absent here in this cartoon, okay. So, now the female will have 2 copies, male will have only 1 copy. So, Morgan reasoned that 1 copy was mutant in that male and the male did not produce the red pigment and therefore it is white. Now, when the gamete forms, the male is going to make Y in one sperm, or like half of the sperm, and then the X chromosome in the other one, and the female will have only 1. And when they are brought together in fertilization, see the females will get 1 copy for sure from the female. So, you have 1 wild type copy, as a result the female will be red, okay, the eyes will be red, and the male here will have 1 copy coming from the female, because you have 1 sperm, right. So, here when the Y comes, then you are going to have this normal allele or the wild type allele coming from the female. So, therefore, in F1 it will be male also will be red color. Now, let us look at the segregation when you go to the next generation. So, this female who got that mutant X chromosome from the male, okay, because male could not have had it. So, to be male it should inherit the Y chromosome from the male, and therefore, this male inherited a good X chromosome from the female, and that is why in F1 it is red color. Now, this mutant X chromosome that came from the male is in this F1 female, and she produces 2 kinds of gametes. One will have the mutant X chromosome, the other one will have the wild type. Now, to make a male you need the Y chromosome, and therefore, you will have 2 kinds of males, one getting the wild type allele from the female, another one getting the mutant allele. And now, you will end up finding in the males you have 1:1 ratio of white color and red color, and you never see red color in the female. So, this sex linked inheritance immediately told the gene for eye color is not just on a chromosome, it is actually on X chromosome. Okay. So, this conclusively proved whatever the cytologists were observing and comparing with the Mendelian inheritance of genes is clearly true, and that genes are on chromosomes. This is something you should know, even if this looks elementary education, but it is worthwhile knowing how we found genes are on chromosomes. Okay, you are not going to tell I am going to do NGS or I am going to do northern hybridization, etc. So, this is how it was found. Is this clear? Okay. So, I will tell you a little bit of history here before we go into the next new idea. The new idea is not that genes are always on 2 different chromosomes and one chromosome has multiple genes. If they have, then what kind of inheritance will be followed. So, that is what is in this slide. So, before going into this, you would be wondering if Morgan struggled hard to find one white male, then now how you have black body, grey body and normal wild type being, vestigial being, how did you get all of this? So, Morgan and his students had no idea at that time how to mutate genes. Okay. Mendel used what is available in nature. So, Morgan searched hard and found one that was found in nature. So, now how do you create variations? So, they decided to mutate, and they did not know what is the nature of gene. So, as a result they did not know how to change that material. If you know the material, you will know how to handle it. So, they kind of tortured the flies, put them in low pressure, high pressure, heat, cold, pluck one leg and or one wing and see what happens. They did all kinds of things, nothing happened. And one of his students, Muller, learned that some physicist called Röntgen had just discovered a thing called X-rays, and they have wavelengths in terms of the distances among atoms in molecules, and gene is probably of that dimension. If it is chromosome, then we are talking about subcellular objects. So, I need to perturb at that level, and these X-rays are probably going to perturb at that level because of its wavelength being at interatomic distances in a molecule. So, they shined X-rays and then they got all kinds of mutant flies. Okay, so, X-rays were the first mutagen used. So, therefore, now we can start with this experiment. Okay, so, now you know why we have these. So, Morgan did an experiment by crossing between one fly strain that had wild type wings but grey abdomen, black and so vestigial is what we are going to say here. So, black is this body color, and when you have the wild type gene, then you call it as B plus, and when you have the normal wing, you call it as vestigial, because in flies as well as in some other organisms, the phenotype is what is eventually given rise to the name. So, body color black. So, wing being small, vestigial wing, they call it as vestigial, that mutant was called vestigial. So, the wild type gene that makes the normal wing is called VG plus. Okay. So, he took one where obviously this is now pure breeding for these two phenotypes. Then they took another one where you have the black body and vestigial wing. Then did a cross, so they got the F1 hybrid where the dominant is showing here, here the dominant is the wild type copy. So, you have one good copy that is enough to make normal wing and grey abdomen. Okay. So, now you have this heterozygous, and we already learned to test cross, and if you do test cross, you should get 1:1 ratio, right, that is what we have learnt earlier. So, instead Morgan got this ratio at the bottom. It was not 1:1 ratio, which would have indicated that each one is following the 3:1 ratio monohybrid wise, and at the same time, so that would have indicated an independent assortment. But if it was not independently assorting, then you should have gotten in this test cross exactly 1:1 ratio, which is equivalent to normal F1 where F1 to F2 3:1 ratio. Okay. So, there you would have selfed, but here you are back crossing, and as a result instead of 3:1 ratio you will get 1:1 ratio. So, he did not get that. So, this made him to think they are probably on the same chromosome. If they are on the same chromosome, we will think that they should follow the rules of monohybrid cross, right. Like if they are together in one chromosome, and do not think crossover, okay, we have not learnt crossover here. So, we do not know what crossover is, we did not learn meiosis. So, we only know this, yeah, this, okay, shape is in one chromosome, color is in another chromosome. Now, if they are in two different things, you will follow 9:3:1 ratio in a normal cross from F1 to F2 without test cross. And if they are on the same chromosome, you will follow 3:1 ratio, right, like for this capital Y and small y coming together here in the various combinations, it is 1:2:1 ratio, and phenotype is 3:1 ratio. So, he did not get either. So, he reasoned that they are probably on the same chromosome, but they have a way of sometimes separating. Probably the chromosomes break and join. Okay. So, that is the idea that Morgan thought of to explain this. okay, evidence for linked genes. So, why did Mendel see something like this when he has used so many different phenotypes? That is simply because fortuitously for him the chromosome size is extremely small, okay. So, he got lucky. So, every phenotype he chose were on different chromosomes because the chromosome size is small. So, maybe if he had looked at 500 different phenotypes, he would have run into this problem. Then you are likely to find 2 genes on the same chromosome. So, here Morgan saw this and then he proposed genes are probably on like multiple genes can be on the same chromosome, but they do not stay there put forever. There seems the chromosomes seem to exchange swap parts, okay. A part of chromosome 1M exchanges equivalent part with chromosome 1P, okay, maternal and paternal. So, that is what I am saying, M and P. So, that is what he proposed, alright, and that is explained here. So, normally so these were the parents and most offsprings will be like this because they are not separating and they are together, or they will be like this. So, this is what normally you would expect, but imagine this happening, crossover, like once the chromosome duplicated, this is let us say chromosome 1M and this is chromosome 1P. After the interphase they are duplicated. Now, the duplicated ones, if they are going to swap parts as you see here, then you can end up producing these 2, right. See, we started with B plus and VG plus. Now, you have a situation where you have B plus and VG, just VG, no plus, and that is because of this swap. So, this was something the cytologists had observed during meiosis, and Mendel proposed something like that can happen and you will have these recombinants, okay. So, this sort of a thing you call as recombinant, like when you are going to get ultimately a phenotype where you have the black bodied fly having normal wing, which is not like this or this, okay. The parental phenotype is normal wing and grey abdomen, but suppose you get normal wing and black abdomen, or black abdomen and normal wing, sorry, that is what I said, and the other one is you have the black body with the normal wing. So, if these things happen, then he called them as recombinant progeny. So, you need to know this, how to identify recombinants. The 2 phenotypes get mixed in the progeny, and because that is essential to think about the next topic in this. So, by proposing that model he could explain how you got this ratio. So, in most of them the swapping did not happen between the P and VG loci, and as a result most of them ended up being like this, parental, resembled either one of the 2 parents, and in some of them where that crossover or recombination happened, you ended up having recombinant offspring, okay. So, in this particular example you can determine the recombination frequency by calculating among the progeny, test cross progeny, you total the progeny you got, which is 2300, and then you look at how many that are recombinants, that is you have a black body with a normal wing or grey body with vestigial wing. When you have that, they do not look like the parent and you total them, and in this particular example that is 391. So, this 391 divided by 2300 times convert to percentage by multiplying 100, then you get 17%. So, the recombination frequency in this case is 17%, okay. So, Muller and few other of Morgan's descendants did not stop there. They proposed if the genes are closer together, then the recombination frequency is likely to be less. The probability of assuming that the crossover, the parts can be swapped anywhere along the length of the chromosome, Now if you take two loci that are farther apart, then the probability a cross happens between the two is higher than when you have chosen them closer together. So that means the recombination frequency is a function of the distance between the two loci. In other words, can I take two different phenotypes and cross them, and based on recombination frequency determine the distance between the two loci on the gene. So this is the basis for genetic mapping. Okay. So the recombination frequency indicates the distance between the two genes on the chromosome. So this way of determining the distance between the two genes is not exactly physical distance. So physical distance we measure by the number of DNA base pairs. This is based on frequency. The frequency is not always directly proportional to the physical length, because due to the chemical nature, like for example the structural uniqueness for different sequences, you could have some parts of the chromosome where recombination readily happens, more readily happens in other loci, you will have variations, but more or less they follow, okay. Except those mutational what we call hot spots where the frequency of recombination is higher than an average locus, except those exceptions mostly this holds good, and this is the basis by which genetic maps are developed. So this kind of tells you, if you take another locus here, this Cn, and then when you do the same experiment as shown in between the black and vestigial, it was determined that in the previous one we saw between B and Vg it is 17%, and then by finding out the distance between these two and these two, people are able to tell Vg is actually on this side, on the left side of black on the chromosome, and it is at this distance. So that we will learn in detail when we go to a genetic cross later, okay. But the main point here is the recombination frequency reflects the distance between different loci on a given chromosome. Okay, so one other thing that you probably overlooked if you were a student like me in the school, but otherwise you may be knowing. So the variations are not we are already seeing that the variation does not mean strictly arising out of new mutations, okay. It can arise due to simply allelic shuffling during crossover, right. Like for example you look at this, right, this individual here and this individual, there was no new mutation that happened in either one of them, no base was changed during DNA replication. It is just that which allele of one gene and which allele of another gene came together because of the crossover generated this variation. This is important because most variations in nature on which nature selection works is based on that. So the variations among us here is not purely due to each one of us generated a new mutation as we began as a zygote. It is simply by which one of your two grandparents during recombination, during gametogenesis in your parents were swapped and brought together, we call that as allelic shuffling, okay. So remember this, in future I might use that word just commonly without explaining. Allelic shuffling creates variation. You do not even need that to create variation, particularly if you consider an organism which has 23 pairs of chromosome. All you need to do is look at this, okay. So now let us look at this. So we are going to have we are going to look at two chromosomes here, okay. So this is chromosome 1 which is duplicated, therefore two sister chromatids are together here, and this is, let us say this came from your mother, the blue color, and this red came from your father, both are duplicated, and now in metaphase they could be like this, purely randomly for both the chromosomes the maternal copy was as it is here on the right side and the paternal copy was on the left side, or it could be this way. For one of them it was on the left side and for the other one it was on the right side, purely by random. There are no selections here. The force that operates here is bringing all chromosomes to the metaphase plate and making sure one of the two homologous chromosomes go to one daughter cell after meiosis 1, while the other one goes to the other daughter cell of the meiosis 1. So, that is all it does. It does not worry about which one of the two allelic chromosomes were taken. So, as shown in this cartoon, you could get this situation. If this happens after meiosis 2, where the sister chromatids are separated, you will end up having combination 1, where you have for both the chromosomes you got the maternal, and combination 2 for both chromosomes you got the maternal, or you could have these. So, you could already have 4 different variations generated in the gamete even without mutations and allelic shuffling coming from crossover. Is this clear? So, you need to remember, because these are all really fundamentals to follow what actually happens in biology. I do not weigh what happens in genetics, because this governs everything else. So, for rest of genetics, what I am going to do is I am going to take one model organism and use a genetic screen done in that organism as an example to understand some of the fundamental concepts. Okay. It could be done using any one of the genetic models. So, I am taking a model where I am familiar with the original literature. So, for obvious reasons it is going to be C. elegans. So, this topic can be taught and understood using Drosophila or Arabidopsis as an example. But in this particular screen you are going to find all different concepts are there in just in that one screen itself. Okay. So, before we go into that, a brief introduction of model organisms. Okay. So, I am going to ask a question. Okay, I will give you one fact and ask a question. Usually when I want to select an organism as a model organism, its genome size smaller the better. Why? Okay, so if I want to look for a gene involved in germ cell development in C. elegans having only 100 million bases versus finding a gene involved in the same gene involved in germ cell development in 3 billion bases, right? I need to screen a minimum of 30 times more individuals after mutagenesis in that the second organism, which is the Homo sapiens. Okay. So, you need to know this. Many times people just say that smaller genome, but they do not understand why smaller genome. And the second one, which is illustrated here, the times. So, here is an adult. Okay, this is about 1 millimeter long, it is not this big as shown in this image. This adult produces embryo which will go through the larval stages at these hours mentioned here, becomes an adult reproducing again in less than 3 days, okay, little more than 2.5 days, but certainly by 3rd day. So, short life cycle, how does that help?
You get multiple generations.
Yes. So, if you go from F1 to F2 and want to find out inheritance pattern and you are looking for recombination frequency, all that in a PhD period, 5 times 365 divided by 3, that many generations you will get. But instead if it is Homo sapiens, you are not even going to have the F1 reaching reproductive maturity in your PhD time. Okay. So, forget about ethical questions, it is a difficult organism to work with. So, those are some of the advantages with C. elegans. There are more, but I am not going to get into all those details, we will learn them as we go. Okay, so this is a necessary information that we need to before we go forward. That is sex determination in C. elegans happens if you have, like in any other organism, if you have any other sexually reproducing organism that we are familiar, 2x means it is a female, it is written hermaphrodites, I will explain in a minute, and if you have x0, there is no Y chromosome. So, it is actually number of X determines here. Okay, people usually say X to autosome ratio. So I will simplify that as number of X chromosome, 2 X chromosome female, 1 X chromosome male. And this X0 arises usually due to defects in meiosis called non-disjunction, where both the homologous chromosomes go into one of the daughters after meiosis 1, therefore the other daughter lacks that chromosome entirely, and the after meiosis 2 the gamete you get will not have the X chromosome if non-disjunction happened with the X chromosome. And so that is how, and if that gamete fuses with a normal gamete having one X chromosome, you get the X0 situation, and those zygotes will eventually end up becoming males. Okay. And interestingly, these females during their sexual maturation, okay, let us say to give a human equivalent, during adolescence when the first germ cell starts to mature, what is going to happen is they are going to enter through spermatogenesis. They are going to follow the male pathway of differentiation, and they become they give rise to sperm, and those sperm produced are stored in a pouch, and then it switches into, after the first 40 germ cells that go through this, roughly 40, it is not fixed 40, then they switch to oogenesis and it behaves like a female. So I said female because her anatomy is a female anatomy. She has the uterus and she has the vulva to lay the embryos out. So bodily she is a female, it is just that she can make sperm as well, but she does that only when she is young. Okay, is this clear? And that is how, because she makes both the gametes, we call her a hermaphrodite. Anatomically she is a female, clear? And it is a sexual reproduction. You have meiosis 1, homologous chromosomes duplicate and separate during meiosis 1, and before that they do have crossover happening, and two gametes will have to fuse to make a new zygote. So it is a perfect sexual reproduction, except that you just imagine you are able to make both gametes. Okay. And so that is the hermaphrodite, and the males are males, normal males like XY males. So anatomically they are differentiated as a male, they have vast difference, make lot of sperm, and they have the copulatory organ to mate with the hermaphrodite, and they do not have uterus or vulva. Okay, so they can't get an egg and fertilize and make a embryo inside their body. So males are like any other sexually reproducing male. So this gives us a big advantage for genetics. So that is one of the reasons why I need to introduce it at this point. So now when the males, if you look at the male testis and you look at the sperm, since males started with X0, now through normal meiosis with no defects, half of the sperm will have X and half of the sperm will have no X, because there is only one X. And therefore when a male mates with a hermaphrodite, 50% of the sperm will have X, 50% will have no X. So as a result, without looking for any meiotic defect, the progeny will be 50% male, 50% hermaphrodite. Okay. So why is this advantageous for genetics? You can do selfing without going through the labor that Mendel went through. All you need to do is take the hermaphrodite, put in one plate. She makes both the gametes. There is no need to cut open the flower and put a cover and take a paintbrush, take pollen, none of that is required. So when you want to cross, you take the male of the right genotype and hermaphrodite of the right genotype, put them together and they will produce a hybrid kids. In next class we will continue from there and we will look at actual experiments and what can be learned from them. So the main point I am driving home is you can learn lot about molecules and their functions without ever actually knowing the DNA sequence or purifying the protein and determining what that protein does. Okay, so that is what we are going to learn at the end of this series of genetics classes.